Today is October 27, 2023. This is the fourth Friday meeting of the OHDSI Phenotype Development Evaluation Workgroup. We meet 2 times a month. We meet on the second Friday and the fourth Friday, and we are OKRs, which we all agreed on in the beginning of the year, are the following. We had 3 main objectives, which is to harden the phenotype development evaluation framework, improve collaboration by enabling the community-wide participation in the phenotype development evaluation, and promote the usage of the OHDSI Phenotype Library. I think we have made good progress on most of them, and we've reflected on this before. I think we made some more progress during the OHDSI Symposium. Mainly, we were able to promote the OHDSI Phenotype Library because it got used in the How Often initiative. So that's a new thing. And also we had we continue to advance the participation in the phenotype development evaluation best practices. I think that those were a few additional things that we can do to take credit towards our key results. So these were the topics that we talked about as agenda, and the main topics that we want to cover today are these items. So we just completed the OHDSI Phenotype Symposium, the Global Symposium 2023. During that, we had a face-to-face meeting. It was a half-day group session. We want to talk about those topics. But before we get into the details, I see some new people who have attended the work group. If this is the first time you're attending the work group and would like to introduce yourself, could you please take this opportunity?
I can go for it. Hi, everyone. I'm Abby. I'm a second-year PhD student in biomedical informatics at Columbia, and I've used the phenotypes from the Phenotype Library in my research, so I'm happy to be here.
Oh, that is so awesome. Thank you. Thank you for saying that, and thank you for using the work that we do, and thank you for coming for this work group. Excellent. Made our day.
Yeah, I can go next. I'm Jeremy. I'm one of the scientific advisors at NICE, and yeah, we're kind of co-project leads with Oxford and Eden. So yeah, wanted to come along to this and yeah, hopefully see some more.
Thank you, Jeremy.
Yeah, we're so happy we're here, Jeremy, because we do want to make sure that what you guys are doing in Eden, in Darwin and Eden gets to the library, so the whole community can use as a resource, particularly phenotype. So that's wonderful, and vice versa, of course.
I can introduce myself. Hi, everybody. My name is Bill Baumgartner. I'm at University of Colorado. I work in Lisa Schilling's group. Lisa's been a long-time member of the OHDSI community, and we're very interested in using the Phenotype Library. And I attended the workshop at the symposium last week, and so I thought I'd come check out the meeting.
Great. Thanks for coming back, Bill. This is great.
Hi, everyone. My name is Guy Livne. I'm the head of Health Informatics, Ministry of Health in Israel. And my goal here with this group is to connect all the information we have in the hospital and all the mind of the physician to be part and contribute to the Phenotype Library and also use it in the research as well.
Fantastic, Guy. And if you're ready to execute the phenotypes in the OHDSI Phenotype Library on your network of data sources, we are ready to enable you and work with you on that.
Thanks. Now we're on a break on everything. It's not crucial in Israel, but I hope as soon as possible to be able to start running it.
I can introduce myself. I'm Christoph Lambert. I'm a professor at University of New Mexico in translational informatics and department of medicine. I've been with OHDSI a long time. I've been doing a lot with positive unlabeled learning to infer the presence of undetected or unrecorded phenotypes, especially in mental health. I work in the mental health work group. I was at the How Often thing, and would like to get things to run and struggle with some phenotypes and things. Also submitted or produced sort of first draft of a phenotype in PTSD. Would like to sort of see that through. So this early time's been hard for me to meet at. So I think I'm nominally part of this group, but have not been really making the meeting. So sorry about that.
So what is translational informatics? Is that like translational medicine for?
Yeah, it's like bioinformatics, chem informatics. The notion of translation is really about, are you ultimately going to have impact on the patient? So
Okay.
bench to bedside type stuff.
Yep.
Sounds like the movement of research to actual clinical care.
Right.
Yeah, that's pretty cool. I wonder if any translational informatics departments there are in the United States. Are you it?
Department, we're a division in medicine. I found there's
okay.
one other division in the world. It's in Japan. So I didn't make up the name. It was my predecessor, and but it's it's kind of unique.
It is interesting. Yeah.
You could call it biomedical, and the closest match would be biomedical informatics, really. For what we.
Translational part is important.
Yeah.
I will adopt this phrase to Israel, the translation informatics. I think it's good for health of everyone.
I don't seem to have access to the chat here, but Joel, I just Skyped you a link to the Science Translational Medicine Journal, which covers a great deal of this kind of stuff, which you might find interesting.
Translational medicine, I know, but translational informatics, I just want to see if they were related.
Same concept.
I'm good with the term translation informatics. I'm not good with the term Swerdale informatics, but let's move on. Avi, I see your hand raised.
No, I was just saying, I was just volunteering. I could go next. So I'm Kavi. I am an assistant professor at Mass General Hospital, and I have been working with the OMOP data model, but this was the first ODC conference, and I'm new to the stack. So it was amazing that it was a great experience to have the stack running. Often you go to hackathon and it's good to have at least a hello world run, right? So it was amazing, and it was what was more amazing was the experience of working with everybody. I think Gautam, you and others on the call, just fabulous to have something running. In the hello world running. My goal right now is to execute the Hoffman study on our 13 million patient database. So that's what is my next step. I work a lot in machine learning in the cardiovascular space. So I want to look at the stack and see how I can contribute or maybe utilize the phenotyping libraries in my work.
That's great. And Kavi introduced himself as somebody who went to medical school, but then decided to get a PhD in computer science. So I think that's the type of phenotypes we want in the phenotype development evaluation work group. So looking forward to collaborating with you, Kavi.
Kavi, I missed. What hospital you at, or what school you at?
Mass General Hospital, Massachusetts General in Boston.
Oh, Massachusetts. Okay.
Oleg, you raised your hand. Go ahead.
Hi, my name is Oleg. I'm also an OHDSI contributor in terms of vocabularies. I'm OHDSI vocabulary tech lead, and I want to do more phenotypes in future. So I came here and participated in the OHDSI phenotyping workshop, this symposium. So I have sort of follow up.
Are you at Columbia, Oleg?
No, at Odysseus.
Odysseus, okay.
Great. Anybody else?
All right.
Oh yeah, good.
Yeah. My name is Chen, and I'm a second-year PhD student at Johns Hopkins University School of Medicine, and I went to the OHDSI Symposium last week, and I worked with Dr. Lampert on developing the phenotype of PTSD there, and I really like the work of developing phenotype. And for my doctoral dissertation, I'm working on to do a network study to reduce low-value care. So that's also, I think, it's really meaningful for me to join this workshop to continue the work.
Awesome. Thank you. Thank you for introducing yourself, and we're looking forward to a long-term collaboration with all of you. Okay, so let's talk about the first agenda item, which is the reflections of the face-to-face meeting. The face-to-face meeting was a workshop, so we all did some work together, and we worked on 7 different clinical ideas using the conceptual framework that this group worked, which is we have a set of 4 steps. We agree on the target clinical idea, we do phenotype development, we do evaluation, and then we do phenotype curation. The focus on the workshop was the third step, which was the phenotype evaluation, and then subsequently maybe phenotype curation. So we divided ourselves into 7 tables, and each table focused on a clinical idea, and the explicit expectations of the workshop is that we'll continue the work and hopefully try to get a paper out of it. So if you are part of any of the tables, could you raise your hands and could you just tell me which group you're in? So Christopher, I see. Christopher, you raised your hand, so go ahead.
Yep, I did PTSD.
Yes. Hello, everyone. Good morning. Good to see everybody again. So I was part of the uveitis kind of ophthalmology table, which I think was really good. You know, we had some content experts there. We had some physicians. We had some data scientists, and then we had kind of an OHDSI expert. Jamie, I think that was you. Thank you. And I think, you know, some good progress.
I don't see him.
Some good progress was made. I think there were some of the pain points were that there was just a lot of different levels of knowledge and expertise around our table. So trying to find that common denominator of involving everyone and having them contribute in a meaningful way was a little difficult. But I think it was nice how the process, you know, the attempt at standardizing the process with, okay, let's start with the definitions, you know, leveraging LLMs, and then go forward, you know, piece by piece. I think that was a nice systematic way of approaching it.
One thing that made our table work better than some of the other tables that I was at was we just set up a Microsoft Teams call and shared a screen, and one person like me drove Atlas as everybody spoke around the table with their ideas. So it wasn't 5 bodies around one screen, but everybody at one screen watching one driver as everybody participated like verbally with each other. And I felt that was just a, you know, like content aside, just process of doing it together, that was actually kind of helpful, because like I'm no ophthalmologist, yet I was surrounded by them. So whenever they gave ideas, I pressed the buttons to support those ideas, and it sort of worked well.
That's awesome. Could you tell us what clinical idea did you guys work on?
And where did you reach? And how can we help in the next steps?
Chris, why don't you go ahead?
Sure. I mean, I think this is largely spearheaded by Brian and Cindy, who were the actual ophthalmologists at our table. But they kind of drilled down from the broad term of uveitis, which is rather non-specific, to really focusing on infectious anterior uveitis. So really being specific with the granularity of that phenotype.
Yeah, it was herpetic and anterior uveitis was the clinical description that we zeroed in on. And I came with a much more general non-infectious uveitis or iridocyclitis definition. And they said that is a very broad non-infectious condition, but for some of the use cases that they were interested in a definition with much more granularity. So we zeroed in on herpetic anterior non-infectious uveitis, and we cooked up a couple definitions, but we have definitely yet to characterize them or run them against Phevaluator.
Did the people in the table achieve a consensus understanding of the clinical idea, the infective anterior chamber uveitis?
I think so. We've wrote down a few things in our document which I no longer know how to access, but we would have to take a look at that.
And you said you had a target clinical idea that you all agreed upon, and you documented as a clinical description?
I think we did. There was some eye rolling at the idea of using large language models to produce it, which led to some discussions about their value and how to go about using that as a starting point, whether you even should. And so we did land on a target clinical idea, but our documentation status. We'd need to dig up the document to see what we wrote down. I can't remember offhand, but we did all agree that this is the thing we were going to study, and we came up with some cohort definitions for it. And I think our definition must be in the doc somewhere. You probably have access to it more quickly than I do, Gavin.
Okay, I'll look at it because I created the shared drive. If you guys have used it, I probably can retrieve that.
I am sure that my general non-infectious uveitis and iridocyclitis definition is in there, but I can't remember if our herpetic anterior uveitis definition is in there or not. I think that wasn't Cindy, it was the other ophthalmologist. What was his name again? Was it
Brian?
Brian, yeah, Brian might have put it in.
And in terms of the step 2, so you developed your own. Did you do a literature search and get?
Not in our 20 minutes together.
Yep, no, understood. And so step 1, step 2, step 3, which was developing the core definitions. You said you built the core definitions from scratch because you did not have something you could leverage from the Phenotype Library. Those definitions, have they been executed on at least one data source and a core diagnostics output reviewed?
We have not done cohort diagnostics, but we have executed them on a couple of J&J sources, a Hopkins source, and Brian does not have Atlas up in his source, but he was able to get the SQL from our Atlas instance in J&J, and he's then learned how to use SQL render and database connector on the spot in order to generate the cohorts on his results schema. I can't remember what his results were, but this was the first time for him to integrate R with database programming and actually got it to do something. So
that's awesome.
Baby steps, but
that's
useful.
Awesome. So you've done step 1, most of it just needs some cleaning up. You've done step 2, which is the phenotype development. You have started the phenotype evaluation. You have to run core diagnostics. You have a network of data sources that you've already kind of agreed upon. You have J&J data sources, you have Brian's data source, and you have Johns Hopkins data source. You've got 3 data partners at the minimum. So do you guys have a timeline? Do you have a follow-up plan to meet together as a group?
Well, Doctor, maybe you want to ask first. Is that an interest, Christopher and Jamie, since you're the two from that table, to continue that work and, characterize these patients and put a paper about how do patients with infectious blah 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 uveitis look like in real world data? Is that intention to continue with that, or you just want to finish? Maybe your final is just submitting a phenotype to the library.
I'm happy to speak to it. I'll also say I think Abby, who's also on this call, was at our table as well, so she might be able to weigh in also. My personal opinion is I probably do not have a lot of interest. Just all cards on the table. I'm not an ophthalmologist. This isn't a phenotype I ever intend on using, and I just have to be smart with my time. I'm happy to weigh in and provide a perspective if that is deemed valuable, but I probably don't intend to invest a lot of time on a specific phenotype.
Yeah, that's very reasonable.
Yeah, I was gonna add in, I think it was a really useful experience, but I have no knowledge of ophthalmology, so I don't know how much use it would be for me to help out.
My goal is to support Brian and Cindy should they want to carry on with the full development and evaluation of this cohort, but my phenotyping bandwidth over the next several weeks is going to be for the non-infectious uveitis or iridocyclitis definition that I submitted to How Often, because it is also a definition being used in a paper that in a manuscript that I'm currently developing that will require a full evaluation report in the long appendix. So that's where my uveitis phenotyping efforts are going to be for the next at least couple of weeks.
Yep.
So maybe Gautam, we take the action to reach out for for Brian and Sydney and see if they have interest in continuing on that. But at least, Jamie, we do have a candidate phenotype algorithms that you guys reached to. So at the minimum, we just finish the step of just looking that they make sense and just putting them in the library if we don't want to go ahead with like a clinical characterization, correct?
Yeah, I think the atlas.odyssey.org, I think we put those definitions on that. I'm just checking right now to see if they got put on there. I think we did put them there, but I think, like you say, getting Brian and Cindy's opinion on where to move with this is probably the best next step.
Do you have the contact information, Jamie? Because you have some interest. Let's
Yeah,
excuse Abby and Christopher. Would you be interested in emailing and asking for a follow-up?
So we can.
Oh, we can do
yeah,
that. All right.
We can do that. We can do that. I have at least Sydney, so we can take that action.
Perfect.
From our side, we at least want to make sure that that phenotype makes it to the library.
Yeah.
Also, I found the links in the Atlas demo. I can send them somewhere, or I don't know.
Awesome. If you would please post that in the group chat, not group chat, the Google Drive shared folder location, I think that would be very useful. Thank you, Abby. All right. Anybody else from this group? Any other comments before we go to the next group, next table? Okay, who wants to go next?
Who's so you have number 1. Abby is the one who has number 1 on her. Yeah, okay. So now we go to number 2. I have on my screen. Hold on. We'll see the full name. Yeah. I think so.
Yeah, it was me.
Yeah, that's you. Yeah, go ahead.
Yeah, we did aspirational pneumonia. So the basic idea was to detect perioperative aspiration, but we merged the 3 ideas from general surgery and pneumonia and aspirational pneumonia. So it was sort of a constructed phenotype. As a result of our work, we actually detected that the existing phenotype in Phenotype Library for perioperative aspiration is not working at all. Like literally it gave 0 patients. And we prepared a clinical description of the phenotype, agreed on that, and we started our literature search. We also constructed some very brief phenotype just to show everyone that it is working and there are patients, but it still requires a lot of work, a lot of follow-up work. So yeah, I'm willing to continue. I can speak for other people, but yeah, I'm willing to continue, and yeah, I'm now working on it actually.
Oh, awesome!
Just reviewing our definition, and yeah, that was it.
So Oleg, did you guys select on like a lead? Are you the lead for your table?
Anna was the lead for our table, since she's the most experienced in phenotype and vocabularies as well. So as of now, I can communicate with her and take this lead from her.
Okay, perfect. So you have a clinical description. It sounds like the table agreed on the clinical idea and documented the clinical description. Is that something that you have put someplace that we have access to?
Yeah, actually, it's in the same folder you shared.
I see. I think I see a general notes.
So yeah, I see it. It's there.
You see it?
I think I see the documents they have. Yeah. So you will continue with Anna, and you guys are close, and we can support also running things. Once you have your final one you want to submit to the Phenotype Library, we can run it. Gautam can add it to the library, which means it's going to generate all the cohort diagnostics result on 7 data sources, and you guys can also look at that and finish your evaluation.
Yeah, perfect. Thank you.
And the core definitions that you are building on, so it sounded like you have built some core definitions. You ran it on a data source that you have access to. You got more than 1 person's, which is more than the number of people that the OHDSI Phenotype Library has for the same aspiration pneumonia as definition. Where is it?
Actually, I need to ask Anna, was it Atlas demo or something like that? It was an open source, and we used this data source.
Okay. Do you have you talked to any data partners now? We can support it from the data source that we have access to. Is there any other data source that you have worked
No,
with?
we don't have. Evan has?
So Evan was on your group, on your table, right?
Yes.
Yeah, he can run things on Stanford right now, Gautam, or
Okay.
he lose.
I don't know.
Okay,
Yeah,
I will
so
talk
we
with him.
Okay, perfect. All right, so your next steps are to finalize your phenotype. So get your clinical description available. Finalize your candidate code definitions, run code diagnostics on one or more data sources, review the results, do a phenotype evaluation, then submit it to the library. That will complete your phenotype development evaluation cycle. After that, you think about clinical application questions that you can ask from those with that phenotype.
Yeah, perfect. Thank you very much. I think I will come on this step, cohort diagnostic step and the real data. So,
Fantastic.
Yeah.
Fantastic. Thank you.
Thank you.
William is next on the hands.
To Bill?
Yeah. So I worked with Hannah Razagi and a few others on the sickle cell anemia phenotypes. And this was Hannah's idea, so I'll try to channel her. But I think the idea was to build phenotypes to distinguish between the homozygous and heterozygous versions of the disease. And we made it through. We made it through the literature search. I mean, we haven't dived into the results, but we were able to run the literature search based on the template you provided. And Evan Minty helped us with the large language model and to produce the clinical description. And so we spent some time kind of going through that. And then we spent some time just comparing some concepts that she had brought and comparing them to the sickle cell anemia phenotypes that I think are currently in the phenotype library, or were in, I don't know, they were in some phenotype library somewhere. So there's still lots of work to be done. When we haven't touched base since last Saturday or whenever it was, but I can reach out to her. And I think the plan is she's affiliated with PedsNet, so I think she was interested in running it on the PedsNet data.
That's great. That's great. Do you have a plan to meet in the near future?
Yeah, I have a plan to reach out to her. I just didn't get to it this week. So.
Perfect. And then we have Guy. Oh, no, not Guy. Sorry, Joel. Joel.
Joel, then Andre, then Guy.
Yeah.
So I think the lead on my table, was that Pavel? Is it for hyperkalemia?
Yes.
Yes.
Yeah.
Okay. Yeah. So he's the lead. I was working with him on it. We did come up with a clinical definition that should be in the folder, I believe. Or else he'll have it. I have to get in touch with him. I need maybe Gotham. You might have some contact information. I'll get in hold of him. It's an interesting phenotype. You know, it looks simple enough. I mean, the one that was originally generated, you know, either had a diagnosis code of hyperkalemia or used a potassium measurement, and probably the potassium measurement is probably going to be as important because it's probably going to be, you know, very low sensitivity with just the condition code. But I checked out the potassium measurement, and it seems like I'm not sure anybody is coming in with a potassium measurement, so we have to see what's going on there. But that would probably be the first thing I need to do is do some diagnostics on that phenotype definition. Otherwise, you know, once we get that settled, then we can start doing the more intensive evaluation of it.
I see. It's an interesting phenotype. I was thinking about it as Pavel was describing, because when I did internal medicine a long time ago, the patients that we were really concerned about was hyperkalemia at the time of discharge. And you would not be able, it's not ethical to discharge somebody who had electrolyte imbalance on the day of. And that's what I remember from my training a lot, more than a decade ago. So it's not, the point here is it was not the reason for the admission, it was a consequence of the admission.
Right. Yeah, that's what Paul was saying. Most of the time it happens during the inpatient stay.
Yeah. Okay. So I'll try to see if I can have the contact information. I'm sure I have his email address. I'll share it with you.
Okay. Thanks.
Andrea?
Yeah, hey, we're working on the definition for anaphylaxis towards the back. Had a couple of colleagues working alongside with me. And I think most of our time we were discussing the current ideas from phenotyping that is started off in, I think, Phenotype February of 2022 and continued the work in 2023. We did a little bit of digging with some OHDSI tools, but none of us had enough familiarity with the tools to make much progress on trying to add extra criteria to the phenotype. And so would like to continue that work, but potentially with a seasoned OHDSI partner that has good access to and familiarity with the OHDSI tools to do some cohort diagnostics.
So what do you think would be the next step? So let's say, what can we do to enable you to continue the journey? We could definitely help you out with experts from OHDSI who are familiar with phenotype development. I could help you, Asa could help you, Joel could help you, somebody else could help you. Given that you have the help, how can we? Do you think you can articulate a plan?
I do wonder, this is probably going to be a phenotype where once we land on a decent definition, I'd really love to add, potential sensitivity, specificity to that. And I really think that that's something that would need chart review. And so that would be probably a step in the relatively near future, because I think that we've already done some exploration, a few steps, rounds of cohort diagnostics. And so at some point soon, I think we'll be looking at manual chart review. Does partners have access to data sets where they have permissions from organizations to do manual chart review?
Some data partners do have access to, and permissions, like I think the hospital-based data partners do, as long as their protocol, as long as they have stated that in the protocol and they have permissions for it. Other data partners, like the data partners that license de-identified data sets, are unable to do so. So you just have to, ask them.
Was this one of the ones that they worked on in Colombia? Was Anna working on this at one point? Because they may have already done some chart review.
For anaphylaxis?
Yeah. I mean, I forget whether this was the one that Anna was interested in, and then she sort of moved on to pneumonia. I forget which. Maybe not. I might be hallucinating.
They did during the COVID ASI work, because this was when anaphylaxis was one of the ASIs. So Anna did dig a little bit in Colombia's, and it wasn't like a full-fledged chart reviews, but she just made sure that we're not making any gross errors during our COVID work. So she looked, but this is like just Colombia, very small scale, during her time there.
Andrea, would you suspect that it's going to be more of a sensitivity issue or a specificity issue?
Well, currently, I think we have like 7 anaphylaxis phenotypes in the library. And so depending on which one we're talking about, it could be both.
Right.
Yeah. I remember from our conversations working with you, Andrea, was the concern that persons who had the code of anaphylaxis were just getting the code because they had history of allergy, and the physicians wanted to provide them an anaphylaxis action plan with like an EpiPen. And so even though they've not actually had anaphylaxis, they would give the code of anaphylaxis on an outpatient basis to get to dispense an EpiPen.
Yeah. Yeah, I agree. I think that depending on which phenotype in the library we're talking about, that's probably a sensitivity error.
Look out
on
and
an
let's do
acute
our
episode. If we wanted to focus instead on a phenotype of patients who have ever had anaphylaxis, so therefore could be at risk of heightened risk of future anaphylactic events, potentially that definition, some of the earlier definitions of anaphylaxis might actually work well.
Gautam, maybe it's on our to do is to clean up the anaphylaxis in the library. I think because we worked on it over 3 years, that's why you have the second version. It's just because it's kind of overworked, and so many people at different time of point. So maybe Gautam, we know where we reached with Andrea last time after Phenotype February, and that scope was actually to the phenotype where events of like active like anaphylaxis that just took place, and not like a prevalent situation. So let's clean up. Let's get to Andrea where we know what that phenotype was after her work last year, or actually early this year, and then take it from there and see what the group wants to do with it. But let's clean it up because I think that caused confusion.
Yeah, that definitely makes sense. I remember spending a few seconds with you, Andrea, and feeling kind of helpless when you expressed that you cannot, getting the number of anaphylaxis options were a lot, and did not know how to solve it at that moment, how to reduce it. And I think one feedback that I take is we do need to reduce the number of options for an end user in the library. And that reduction should be based on some rubric. And that rubric could be, at the bare minimum, it could be these things that we talked about at the beginning of the call, which is like cohort hygiene. Like, have they, has, and then there's always the idea of the operating characteristics as a second level.
Yeah, and maybe something like, all the cohorts are basically named anaphylaxis. If there was if some of the things in the library are in progress, maybe an extra little tag on the name could help people know that they're not finalized or something like that.
Yeah. There's a lot of administrative things to be done there.
Yeah, it's 9:46. We're gonna move for.
Yeah.
So we have Guy and
Abhi.
Yeah, and Abhi again. Yep. Go ahead, Guy.
Yeah, I was in Andrea's table, and I can say that we do need someone from OHDSI to help us with the tool. We were trying to eventually decide on a specific code and try to use the code diagnosis, but we actually didn't get so far. I'm really willing to continue with this, working as a group, and I think I can also be able, while understanding more the step forward, to add some clinicians from the hospital board. So I think it can be helpful to help the finalize the definition of each of the sub-anaphylaxis phenotypes.
Yeah. Do you have any?
And I also have access to data. I need to understand better what is needed in order to do the validation. Yeah, I do have access to data from 6 different hospitals.
Abhi?
Yeah, I just had a quick question. Because I'm new, if you could just give like in a 1 minute, like why are we developing these phenotypes? How we choose them, and roughly like what should I be looking at to catch up?
Yeah, absolutely. So So I'll answer. So why do we do this? So if to do good quality research in observational data, we need to make sure that we reduce the measurement errors as much as possible. Because if we have measurement errors, then our results are not reliable or trustable. There's very few ways that we can evaluate if the cohort that we are studying is truly representing the target clinical idea. So we have these tools like cohort diagnostics and fee evaluator that does a reasonable job. And then there are other techniques like chart reviews and so on that also does a reasonably good job. Some of them are feasible, others are not feasible. And so there's lots of challenges here. So essentially the first order problem that we are trying to solve is we just don't want to build a definition and just use it blindly. We need to know its measurement errors so that we can try to fix it. And even if we can't fix it, then we have to make the decision whether we want to accept it. Because those knowing upfront that there are some potential sources of errors and that way bias is good practice. So that's the fundamental philosophical idea. Now, in terms of how we selected the 7 clinical idea, that's just basically people volunteering and say, this is what we're interested in. And because it was a workshop and we had to do something, it's very hard to do a lot of things in a very short amount of time. So I exchanged some emails with the people who signed up to the work group and said, what are you interested in? Can I help you in any way? And some of them said upfront that we're interested in this one or that one. And then I tried to say, here's a few things that are in the library that might accelerate it. Like we saw, doing all 4 steps takes days, if sometimes even weeks. And to do it in like 2 hours in a group setting, that was a challenge. But the point of that was just to trigger this conversation so that over the next few weeks or maybe a months, we can complete it. That was the idea, Kavi.
And Kavi, we as a working group, we have main goals. One is to build that phenotype library for the community, so people like Abby doing her research can go into a resource, and instead of starting to phenotype everything from scratch, can find phenotype algorithm that she can use in her clinical research or even methodological research. Now that library needs content and need people to continuously keep building. So we take advantage of times we meet for people who are interested either on a clinical idea perspective, just like Christopher and others. And some people are just interested in knowing from informatics perspective, knowing how to do that, how to select codes, how to build algorithms, how to use tools to help them to do that. So we're trying to kind of help everybody while building phenotypes. One, they get used, they get training on how to do that on their own data source using OHDSI tools, and also clinicians get to get informatics help to get the clinical knowledge into a phenotype algorithm, because that's where most of the clinical knowledge need to go into research, because that's where you define things and then you run an analysis. Usually in OHDSI, at least it's standardized. So that's where we try to help people get to do that. So we want to build up the content of the library. So we use all the time we have together to increase the content. We want to help the clinicians to get some informatics help, and we want to get the informatician to understand how to use our tools in relation to phenotyping.
And I'll add one more topic. So this is a lot of work, and most people are challenged to do this type of work because where does it end? And so one of the things that we are trying to do is to say this type of work is actually publishable as an independent body of work. And so we started out the workshop with examples from Joel and Jill, where they said Joel made the point that just the body of work that is phenotype development evaluation by itself is publishable. And Jill made the point that you can also do a clinical application paper where the phenotype development evaluation is part of it or as its thing. So essentially, this body of work, anaphylaxis, anterior chamber uveitis, infective, in aspiration pneumonia, we believe that is independently scientifically publishable in a peer-reviewed scientific journal. And so we as a community would like to do that because that I think will improve the quality of observational research.
That's awesome. Thank you, both for this answer. So cohort diagnostics and fee evaluator sound very exciting. I'll look them up. But is there like a document, let's say I want to build a cohort, right? So now it's good, like cohort we're building in general for everybody, and not that we have a particular study in mind, or if someone has in mind, they can propose one. So I noticed that there are 250 phenotypes already over there, and so if one has to build a phenotype, is there like a dummies, document like, okay, why are we doing this? How are we doing it? That I could use?
Yeah. So there are several bodies of work. There is a book called the Book of Odyssey, which I think is the starting place for most people, and that introduces the concept of what is a cohort and what are the tools that are supporting it. It also talks about how to install the tools and also how to use the tools. Just so that's the basic introduction. Then there is some tutorials that some of them are recorded on YouTube that talks about how do you model a target clinical idea using like the tools like Atlas. So what is an entry event criteria? What is the inclusion rule? What is the exit criteria, and so on. Now it goes to some depth, but Atlas is a pretty complex tool with a lot of sophisticated temporal rules that it brings forward. And so it takes some time to develop proficiency in it. So what we do in a program like Phenotype February is when somebody who's reached the medium level proficiency but wants to take it to the next level, or at least has the basic level proficiency, we try to spend time with them to solve their modeling question, like how do they model a clinical idea, what different ways. So we do that, and those are recorded. So if you look at Phenotype February, some of these are discussed live and are recorded. This is a time-consuming process, training. And when you go to the advanced level, there's a lot of tips and tricks and so on. That's like your ninja mode. It takes time to get to that level.
Got it. Thanks. I'll just document my journey, and thanks for pointing me. I'll be going through this. Maybe if it helps, I'll just document it in one place, and maybe I don't know, it might be helpful.
No, that's fantastic. There's actually nobody who's written a paper on Atlas itself.
Yeah, there's papers for Atlas, but not like just Atlas as a phenotyping tool. Yeah.
Could I talk a bit about the PTSD effort?
Yes.
Yeah.
I got to run to my 8 o'clock meeting.
If you are in a safe place only, please.
Yeah, I'm driving, but that's okay. So the we narrowed it not just to PTSD, but pharmacologically treated by the two FDA-approved compounds, sertraline and paroxetine. I have the working in the demo.odyssey.org. It's running in Atlas, so I think it's in terms of computationally, it works, it runs, although on my own Atlas instance, it dies on my bigger database, and I wish there was a way to see like log messages of why the generate fails. So I guess just guidance on, where to go from here. I mean, we could edit more our phenotype description that sort of, was kicked off with ChatGPT in the session. But, just what would be, should we just post it to the forums next and try to, and it's you guys who do cohort diagnostics, or there's a way for us to do it?
Yeah, so I'll try to answer. So, I just pulled up a code definition that I think it was what you built, Christoph, and I think I can review it and just make sure there is no like errors that might explain why it did not run on a large data source. I'm happy to run it on another data source that I have access to see if it actually runs. So once we confirm the hygiene part of the code definitions, then I can create the code diagnostic package for you so that you can run it as a study, and then you can also ask others in the Odyssey network to run it for you. So you can get like a composite of several people's data sources. We can then deploy it on data.odyssey.org and create a PTSD app, Shiny app, which we can review together. And once we do it, we'll look at sources of errors and we'll document that in the forum post. You'll be the lead on it, document it. and then we'll take that into the library. And once it's in the library, you can cite it and write a paper on it.
Very good. Okay. We're most interested in like treatment pathways. You know,
Yep.
so like you start on these FDA approved drugs, and you know what's the sequence after that. So be good to get it validated and try to run that tool.
Great. So my first step is I'm looking at your definition. I'll just look at it from a hygiene perspective and see if it'll run. And if it runs, then I'll create a code diagnostic package for you to run on your data source. I'll put it on OHDSI studies repository, and then it's up to you to like ask the network to run it. And if you get many people running it, we'll just put it in a Shiny app together and then review it as a group.
Okay, thank you.
So Gautam, we have to close, but may I suggest for our next we're meeting in 2 weeks.
Yeah.
So it looks like we have at least candidate cohort definition, phenotype algorithm for PTSD from Christoph table, and then the infectious uveitis from Jamie and Christopher.
We have aspiration pneumonia.
And we have aspiration pneumonia, and then we have hyperkalemia. At least after Jewel look at the measurements and just make sure we're not missing some values. So maybe in our next meeting we have them in a cohort diagnostic instance, and just go over them quickly with the group from a cohort diagnostic perspective, and then see the next steps for just evaluated. So we at least make sure that we have the minimum step for them to be submitted to the library, or at least finalize them as so. So instead of leaving everybody doing their work, we have candidate phenotypes for those, so we can take the time next in 2 weeks and just look at them in cohort diagnostics and make sure we have the inputs for evaluated. Does that make sense?
Yep.
So that would be our agenda in the next 2 weeks, just so we can follow up, finish the work that was initiated in the workshop from a phenotyping perspective.
Okay.
That sounds great.
All right. Thank you, everybody. We are on time. I hope this was a good discussion, and I'm looking forward to talking to you guys again in 2 weeks.
Thank you.
Thank you.
Thank you. Bye. Take care.
Bye.